This table highlights the effect of inheritance of either the dominant wild-type secretor gene or the recessive mutated form or deletion of the gene. Note the red cell antigen expression is only affected in those who are homozygous recessive for the H gene. This table gives an overview of the population frequencies for different blood groups. This is important when considering cross-matching in patients of different ethnicities. For example, B and AB blood groups are much more common in Japanese compared to UK native populations. The differences between the Scottish and UK frequencies highlights the potential for regional variation. So A, B, and O are three main carbohydrate-based blood groups. All are dependent on formation of H substance, the formation of which is dependent upon the expression and activity of the enzyme 1,2-fucosyltransferase, which adds a fucose to a precursor carbohydrate molecule. Enzymic addition of different sugars results in formation of group A or group B. H substance in group O remains unaltered. So now we are going to examine some of the subgroups of A which are caused by a dysfunctional transferase enzyme gene and results in quantifiable reduction in A antigen levels. Similarly, blood group B has subgroups. The subgroups of A are A1, A2, A3, and AN, AFIN, AL, AX, AIT, and AM. Some of these are specific to populations, such as AFIN, which is present in the Finnish population. Importantly, each exhibit their own serological characteristics, making interpretation challenging. 80% of group A individuals are A1, almost all the rest being A2. This slide shows variation in A antigen expression per micrometer squared across the red blood cell surface. A2 have less A antigen on the cell surface, therefore the person who is A2 can form anti-A1, i.e. antibody that reacts with the A1 group. In rare cases, this can cause a transfusion reaction. Anti-A1 and A2 are detected by the lectin Dolichos bifluus, which reacts with A1 cells, but not A2. This slide is simply a diagrammatic representation of the amount of A antigens and some different subgroups. The policy at 9 wells is anti-A1 antibody samples are cross-matched with A1 negative cells. This is not the case everywhere, but this policy reduces the chance further of a transfusion reaction. Most examples will only agglutinate up to 25 degrees C, therefore are of no clinical significance. However, they can cause grouping anomalies. In some rarer cases, they're active at 30 and 37 degrees C. Regardless of the thermal amplitude, all patients with anti-A1 antibodies will be cross-matched with A1 negative cells. Each subgroup has a different serological characteristic. So taking AX as an example, AX efficiently absorbs and elutes anti-A1, which means there is insufficient A antigen on cells to cause agglutination. However, anti-A1 antibodies will bind to A antigens on cells, so when an eluate is performed, that is, chemical breaking down of red cells to release the antigen, you would find anti-A1 antibody is bound to the red cells. Consequently, AX plasma contains anti-A1 antibodies. AX is also agglutinated by anti-AB, which is a bigger multivalent antibody. AX plasma contains anti-A1, which can be used to determine the subgroup of A. The frequency of AX is quite low, at 1 in 40,000. So to summarise some key points, the ABO blood group system is the classification of human blood based on the inherited properties of red blood cells as determined by the presence or absence of the antigens A and B, which are carried on the surface of the red cells. A and B are codominant, so a person may thus have type A, type B, or type AB blood. Someone with blood group O have red cells that do not express A or B. A and B antigens are carbohydrates and built sequentially by the activity of enzymes that are produced products of the H allele and A and B alleles. Homozygous recessive individuals for the H allele cannot form A or B antigens. And finally, subgroups of group A and B are of clinical significance.